Spermatogenesis. Spermatogenesis is the production of sperm from the primordial germ cells. Once the vertebrate primordial germ cells arrive at the genital ridge of male embryos, they become incorporated into sex coats. They remain there until maturity, at which time these coats hollow out to form the seminiferous tubules, and the epithelium of the tubules differentiate into the Sertoli cells. During his lifetime, a human male can produce 10 raised to 12 or 10 raised to 13 gametes. So the spermatogenic cells are bound to the Sertoli cells by the N-cadherin molecule on their respective cell surface and by galactosyl transferase molecule on the spermatogenic cells that bind the receptor on the Sertoli cells. So the Sertoli cells nourish and protect the developing sperm cells, and spermatogenesis, the developmental pathway from spermatogonial stem cell to mature gamete, occurs in the rhesus of Sertoli cells. The process by which the PGCs generate sperm have been studied in detail in several organisms, but we will focus here on spermatogenesis in mammals in particular. So after reaching the gonad, the PGCs divide to form type A1 spermatogonia. These cells are smaller than the PGCs and are characterized by the ovoid nucleus that contains chromatin associated with the nuclear membrane. The A1 spermatogonia are found adjacent to the other basement membranes of the sex coats. At maturity, these spermatogonia are thought to divide so as to make another type A1 spermatogonium as well as second paler type of cells, the type A2 spermatogonium. Thus, each type A1 spermatogonium is a sperm cell capable of regenerating itself as well as producing a new cell type. The A2 spermatogonia divide to produce A3 spermatogonia, which then beget the type A4 spermatogonia. It is possible that each of the type A spermatogonia are stem cells capable of self-renewal. A A4 spermatogonium has three options. Can form either A4 spermatogonium, that means self-renewal. It can undergo cell death with the help of apoptosis, or it can differentiate into the first committed stem cell, the intermediate spermatogonium. Then intermediate spermatogonia are committed to become spermatozoa, and they mitotically divide once to form the type B spermatogonia. These cells are the precursors to the spermatocytes and are the last cells that undergo mitosis. These cells divide once to generate the primary spermatocytes, the cells that enter meiosis. It is not known what causes the spermatogonia to take the path followed, differentiation rather than self-renewal, or it is known what stimulates the cell to enter meiotic rather than mitotic division. So you find that during these spermatogonial divisions, cytokinesis is not complete. Rather, the cells form a syncytium whereby each cell communicates with the other via cytoplasmic bridges about 1 μm in diameter. The successive divisions produce clones of interconnected cells and become ions and molecules readily pass through these intercellular bridges. Each cohort matures synchronously. Then each primary spermatocyte undergoes the first meiotic division to yield a pair of secondary spermatocytes which complete the second division of meiosis. The haploid cells formed are called spermatids, and they are still connected to each other through their cytoplasmic bridges. The spermatids that are connected in this manner have haploid nuclei but are functionally diploid, since the gene product made in one cell can readily diffuse into the cytoplasm of its neighbors. During the divisions from type A1 spermatogonium to spermatid, the cells move further and further away from the basement membrane of the seminiferous tubule and closer to its lumen. So thus each type of cell can be found in a particular type layer of tubule. The spermatids are located at the border of the lumen, and here they lost their cytoplasmic connections and differentiate into sperm cells. In human, the progression from spermatogonial stem cells to mature sperm takes 65 days. Then spermiogenesis, the haploid spermatid is a round, unflagellated cell that looks nothing like a mature vertebrate sperm. Next step in spermaturation then is spermiogenesis or spermatolysis, the differentiation of the sperm cell. For fertilization to occur, the sperm has to meet and bind with the egg. And spermiogenesis differentiates the sperm for these functions of motility and interaction. The process of mammalian sperm differentiation can be seen. So the first step involved the construction of acrosomal vesicles from the Golgi apparatus. The acrosome forms a cap that covers the sperm nucleus. As the cap is formed, the nucleus rotates so that acrosomal cap will then be facing the basal membrane of the seminiferous tubule. This rotation is necessary because the flagellum is beginning to form from the centriole on the other side of the nucleus, and this flagellum will extend into the lumen. So during the last stage of spermiogenesis, the nucleus flattens and condenses, and remaining cytoplasm, the cytoplasmic droplet is jettisoned, and the mitochondria from the ring around the base of the flagellum. The resulting sperm then enter the lumen of the tubule. When in the mouse, and the mitochondria form the ring around the base of the flagellum. The resulting sperm then enter the lumen of the tubule. So in the mouse, the entire development from stem cell to spermatozoon takes 34.5 days. The spermatogonial stages last 8 days, meiosis lasts 13 days, and spermiogenesis takes up another 13.5 days. In humans, this development takes nearly twice as long to complete because the type A1 spermatogonia are stem cells. Spermatogenesis can occur continuously. Each day, some 100 million sperms are made in each human testicle, and each ejaculation releases 200 million sperm. So unused sperm are either reabsorbed or passed out of the body in urine. Then next topic is about oogenesis. So oogenic meiosis. Oogenesis, the differentiation of ovum, differs from spermatogenesis in several ways. Whereas the gamete formed by spermatogenesis is essentially a motile nucleus, the gamete formed by oogenesis contains all the factors needed to initiate and maintain metabolism and development. Therefore, in addition to forming a haploid nucleus, oogenesis also builds up a store of cytoplasmic enzymes, mRNAs, organelles, and metabolic substrates. While the sperm becomes differentiated for motility, the oocyte develops a remarkably complex cytoplasm. The mechanisms of oogenesis vary more than those of spermatogenesis. This difference should not be surprising since the patterns of reproduction vary so greatly among species. In some species, the sea urchins and frogs, the female routinely produces hundreds or thousands of eggs at a time, whereas in other species, just humans and most mammals, only a few eggs are produced during the lifetime of an individual. In those species that produce thousands of ova, the oogonia are self-renewing stem cells that endure the lifetime of the organism. So, in these species that produce fewer eggs, the oogonia divide to form a limited number of egg precursor cells. The mechanism of oogenesis vary more than in those of spermatogenesis. So this difference should not be surprising, we mentioned. Then other species such as humans and most mammals, only a few eggs are produced during the lifetime of an individual. In those species, that produce thousands of ova. The oogonia are self-renewing stem cells that endure for the lifetime of the organism. In those species that produce fewer eggs, the oogonia divide to form limited number of egg precursor cells. In humans, the 1,000 or so oogonia divide rapidly from the 2nd to the 7th month of gestation to form roughly 7 million germ cells. After the 7th month of embryonic development, however, the number of germ cells drop and most oogonia die during this period, while the remaining oogonia enter prophase of the first meiotic division. These later cells, called the primary oocytes, progress through the first meiotic prophase until the diplotin stage. They undergo first meiotic prophase until the diplotin stage, at which point they are maintained until puberty. So they are stuck in the first meiotic prophase stage until the diplotin stage. With the onset of adolescence, groups of oocytes periodically resume meiosis. Most part of meiosis is begun in the embryo, and the signal to resume meiosis is not given until roughly 12 years later. In fact, some oocytes are maintained in meiotic prophase for nearly 50 years. Primary oocytes continue to die even after birth. Of the millions of primary oocytes present at birth, only about 400 mature during a woman's lifetime. Then, oogenic meiosis also differs from spermatogenesis meiosis in its placement of the metaphase plate. When the primary oocyte divides, the nucleus of the oocyte, called the germinal vesicle, breaks down and the metaphase spindle migrates to the periphery of the cell. At telophase, one of the two daughter cells contain hardly any cytoplasm, whereas the other cell has nearly the entire volume of cellular constituents. A smaller cell is called first polar body, and the larger cell is referred to as the secondary oocyte. During the second division of meiosis, a similar unequal cytokinesis takes place. Most of the cytoplasm is retained by the mature egg, and the second polar body receives little more than a haploid nucleus. This oogenic meiosis serves to conserve the volume of oocyte cytoplasm in a single cell rather than splitting it equally among 4 progeny. So, in a few species of mammals, meiosis is severely modified such that the resulting gamete is diploid and need not be fertilized to develop. Such animals are said to be parthenogenetic. We know that. In the fly Drosophila melanogaster, one of the polar bodies acts as a sperm and fertilizes the oocyte after the second meiotic division. In other insects, such as Moraba virgo and the lizard Nemidophorus uniparens, the oogonia double their chromosome number before meiosis so that halving of chromosomes restore the diploid number. The germ cells of the grasshopper Cyanocellus surinamensis disperse with meiosis altogether, forming diploid ova by 2 meiotic divisions. In the preceding example, the species consist entirely of females. In other species, haploid parthenogenesis is widely used not only as a means of reproduction but also as a mechanism of sex determination. In the Hymenoptera, that means bees, wasps, and ants, unfertilized eggs develop into males, whereas fertilized eggs, being diploid, develop into females. Unfertilized eggs develop into males, whereas fertilized eggs, being diploid, develop into females. So, In case of bees, wasps, and ants, all those unfertilized eggs are actually males, and all those fertilized eggs are females. The haploid males are able to produce sperm by abandoning the first meiotic division, thereby forming two sperm cells during second meiosis. And maturation of the oocyte in amphibians. The egg is responsible for initiating and directing development, and in some species, fertilization is not even necessary. The accumulated material in the oocyte cytoplasm include energy sources and organelles, the yolk and mitochondria, the enzymes and precursors of DNA, RNA, and protein synthesis, stored messenger RNA, structured protein, and morphogenetic regulatory functions that control early embryogenesis. A partial catalogue of the material stored in the oocyte cytoplasm is important. Most of this accumulation take place during meiotic prophase 1, and this stage is often subdivided into pre-vitellogenic, that means before yolk formation in Greek, and vitellogenic, yolk forming phases. So eggs of fishes and the amphibians are derived from a stem cell oogonia population that can generate a new cohort of oocytes each year. In the frog Rana pipens, oogenesis lasts 3 years. During the first 2 years, the oocyte increases its size very gradually. During the third year, however, rapid accumulation of yolk in the oocyte causes the egg to swell to its characteristically large size. Eggs mature in early batches, the first cohort maturing shortly after metamorphosis. The next group matures a year later. So vitellogenesis occurs when the oocyte reaches the diploidine stage of meiotic prophase. Yolk is not a single substance but a mixture of materials used for embryonic nutrition. The major yolk component is 470 kilodalton protein called vitellogenin. It is not made in frog oocyte, as are the major yolk proteins of organisms such as annelids and crayfishes. It is synthesized in the liver and carried by the bloodstream to the ovary. This large protein passes from the follicle cells of the ovary and is incorporated into the oocyte by micropinocytosis, the pinching off of membrane-bound vesicles at the base of microvilli. In the mature oocyte, vitellogenin is split into two smaller proteins, the heavily phosphorylated phosphitine and the lipoprotein lipovitellin. These two proteins are packaged together into membrane-bound yolk platelets. Then glycogen granules and the lipochondrial inclusions store the carbohydrate and the lipid components of the yolk. Most eggs are highly asymmetrical, and it is during oogenesis that the animal-vegetal axis of the egg is specified. Gerhardt and Danilchik have shown that although the concentration of yolk in xenopus oocytes increases nearly tenfold as one goes from the animal to the vegetal poles of the mature egg. Next, we are going to learn about the oogenesis in mammals. Ovulation of the mammalian egg follows one of two basic patterns depending on the species. One type of ovulation is stimulated by the physical act of intercourse itself. Then physical stimulation of the cervix triggers the release of gonadotropins from the pituitary. These gonadotropins signal the egg to resume meiosis and initiate the events of expelling the ovum from the ovary. This method ensures that most copulations lead to fertilized ova, and animals that utilize this method of ovulation, rabbits and minks, have a reputation for procreative process. Then most mammals, however, has a periodic type of ovulation. The female ovulation only at ovulates. The female ovulates only at specific times of the year called estrus in its English equivalent heat. In these cases, environmental cues, most notably the amount and type of light during the day, stimulate the hypothalamus to release gonadotropin releasing factor. This factor stimulates the pituitary to release its gonadotropins, follicle stimulating hormone. Follicle stimulating hormone. That means FSH in short. And luteinizing hormone LH, which causes the follicle cells to proliferate and separate estrogen. Secrete estrogen. The estrogen subsequently enters certain neurons and evokes the pattern of mating behavior characteristics of the species. So gonadotropins also stimulate follicular growth and the initiation of ovulation. Thus estrus and ovulation occur close together. So humans have a variation on the theme of periodic ovulation, although human females have a cyclic ovulation averaging about 29.5 days and no definitely yearly estrus. Most of human reproductive physiology is shared with other primates. The characteristic primate periodicity in maturing and releasing ova is called the menstrual cycle because it entails the periodic shedding of blood and cellular debris from the uterus at monthly intervals. The menstrual cycle represents the integration of the three very different activities. The ovarian cycle, the function of which is to mature and release an oocyte. Then the uterine cycle, the function of which is to provide the appropriate environment for the development of blastocyst to implant. And the cervical cycle, the function of which is to allow sperm to enter the female reproductive tract only at the appropriate time. These three functions are integrated through the hormones of the pituitary, hypothalamus, and ovary. Then, the majority of the oocytes within the adult human ovary are maintained in the prolonged diplotin stage of the first meiotic prophase, often referred to as a dictate stage. Dictate, D-I-C-T-Y-A-T-E, dictate stage. Diplotin stage of first meiotic prophase. Diplotin stage of first meiotic prophase. Each oocyte is enveloped by a primordial follicle constituting of a single layer of epithelial granulosa cells and a lesser organized layer of mesenchymal thecal cells, which are the primordial follicle constituting a single layer of epithelial granulosa cells and a lesser organized layer of mesenchymal thecal cells. So periodically a group of primordial follicle enter a stage of follicular growth. During this time the oocyte undergoes a 500-fold increase in volume corresponding to an increase in oocyte diameter from 10 μm in a primordial follicle to 80 μm in a fully developed follicle. So concomitant with the oocyte growth is an increase in the number of follicular granulosa cells which form concentric layers about the oocyte. This proliferation of granulosa cells is mediated by a paracrine factor GDF9, a member of the TGF-beta family. So throughout this growth period, the oocyte remains in the dictate state. The fully grown follicle, thus containing a large oocyte surrounded by several layers of granulosa cells. Many of the cells will stay with the ovulated egg, forming the cumulus which surrounds the egg in the oviduct. In addition, during the growth of the follicle, the antrum or cavity forms which become filled with complex mixture of proteins, hormones, CAMP and other molecules. At any given time, a small group of follicles is maturing. However, after progressing to a more mature stages, most oocytes and their follicles die. To survive, the follicle must find a source of gonadotropic hormones and catching the wave at the right time must ride it until it peaks. Thus, for oocyte maturation to occur, the follicle needs to be at a certain stage of development when the waves of gonadotropin rises. So, day 1 of the menstrual cycle is considered to be the first day of bleeding. So, this bleeding from the vagina represents the sliding off of extra uterine tissue and blood vessels that could have aided the implantation of the blastocyst. In the first part of the cycle, called the proliferative or follicular phase, the pituitary gland starts secreting an increasingly large amount of FSH. The group of maturing follicles which have already undergone some development respond to this hormone by further growth and cellular proliferation. FSH also induces the formation of LH receptors on the granulosa cells. Shortly after this period of initial follicle growth, the pituitary begins secreting LH. In response to LH, the meiotic block is broken. So the nuclear membrane of competent oocytes break down and the chromosomes assemble to undergo the first meiotic division. One set of chromosomes is kept inside the oocyte and the other is given to the small polar body. Both are encased by the zona pellucida, which has been synthesized by the growing oocyte. So after that, what is happening? It is in this stage that the egg will be ovulated. So the two gonadotropins acting together cause the follicle cells to produce increased amounts of estrogen, which has at least 5 major activities in regulating the further progression of the menstrual cycle. It causes the uterine mucosa to begin its proliferation and to become enriched with blood vessels. It causes the cervical mucus to thin, thereby permitting sperm to enter the inner portions of the reproductive tract. It causes an increase in the number of FSH receptors on the follicular granulosa cells, while causing the pituitary to lower its FSH production. It also stimulates the granulosa cells to secrete the peptide hormone inhibin, which also suppresses pituitary FSH secretion. At low concentrations, it inhibits LH production, but at high concentrations, it stimulates it. At very high concentrations and over long durations, estrogen interacts with the hypothalamus, causing it to secrete gonadotropin releasing factor. As estrogen levels increase as a result of follicular production, FSH levels decline. LH levels, however, continue to rise as more estrogen is secreted. As estrogens continue to be made day 7 to 10, the granulosa cells continue to grow. Starting at day 10, estrogen secretion rises sharply. This rise is followed at mid-cycle by an enormous surge of LH and a smaller burst of FSH. So experiments with female monkeys have shown that exposure of the hypothalamus to greater than 200 pg of estrogen per milliliter of blood for more than 50 hours results in the hypothalamic secretion of gonadotropin releasing factor. This factor causes the subsequent release of FSH and LH from the pituitary within 10 to 12 hours after the gonadotropin peak. The egg is ovulated. So although the detailed mechanism of ovulation is not yet known, the physical expulsion of the mature oocyte from the follicle appears to be due to an LH-induced increase in the collagenase, plasminogen activator and prostaglandin within the follicle. The mRNA for plasminogen activator has been dominant in the oocyte cytoplasm. LH causes this message to be polyadenylated and translated into its powerful protease. So prostaglandins may cause localized contractions in the smooth muscles in the ovary and may also increase the flow of water from the ovarian capillaries. If ovarian prostaglandin synthesis is inhibited, ovulation does not take place. In addition to the prostaglandin-induced pressure, collagenase and plasminogen activator protease loosen and digest the extracellular matrix of the follicle. Then the result of LH then would be increased follicular pressure coupled with the degradation of the follicle wall. A hole would be digested through which the ovum would burst. Following ovulation, the luteal phase of menstrual cycle begins. The remaining cells of the luteal follicle under the continued influence of LH become the corpus luteum. They are able to respond to this LH because The surge in FSH stimulates them to develop even more LH receptors. The corpus luteum secretes some estrogens, but its predominant secretion is progesterone. The steroid hormone stimulates to the uterus, where it completes the job of preparing the uterine tissue for blastocyst implantation, stimulating the growth of the uterine wall and its blood vessels. So, blocking the progesterone receptor with the synthetic steroid mifepristone stops the uterine wall from thickening and prevents the implantation of blastocyst in the uterus. So, progesterone also inhibits the production of FSH, thereby preventing the maturation of any more follicles to ova. For this reason, such a combination of estrogen and progesterone has been used in birth control pills. The growth and maturation of new ova are prevented so long as FSH is inhibited. If the ovum is not fertilized, the corpus luteum degenerates. Progesterone secretion ceases. The uterine wall is rooted off. With a decline in serum progesterone levels, the pituitary secretes FSH again and the cycle is renewed. However, if fertilization occurs, the trophoblast secretes a new hormone, luteotropin, which causes the corpus luteum to remain active and serum progesterone levels to remain high. Thus, the menstrual cycle enables the periodic maturation and ovulation of human ova and allows the uterus to periodically develop into an organ capable of maturing a developing organism. Developing organism for 9 months.